and I mentioned that we would have a computer lab room, so that room is 001, so it's, in the basement of this building, 001. So this Friday, that's where we meet. I know the football team and the soccer team are traveling. There may be others. If you fall in that category, let me know. Basically, what I'm going to try and do for those folks is set up some kind of after-hours group meeting to just go through this exact same thing we're doing in class. Just because it's the start of a project, so I don't want everybody kind of feel comfortable with what we're going to do. And so on Friday, you're certainly welcome. Even though we're in the computer lab, it will seem silly, but you're certainly welcome to use your own laptop. The reason why we go to the computer lab is that way I'm 100% sure that everybody has access. There's still those of us out there that like the good old desktop. And so for those individuals, they may not have something to tote around with them, but I want everybody to be able to work on their stuff. So point being, I don't care what you decide to do the searches on, but we'll work through that first worksheet. Also, I had you print off the baloney detection kit and the APA sheet. Probably we won't get to that. And we'll pick it up on Friday at the beginning of the class period. Now, the APA sheet, you can start working on it. The point to that, so at the very top, you'll notice it says you must turn this in before you turn in some kind of paper, something to that effect. Does not apply to us. Dr. Zell uses that in her, so obviously she created it, but she uses it for her students as a quiz. But I've gotten permission to provide the answers to you. So basically, we're going to simply use it as practice. Because the more we do it, the more we work through it, the easier this will be. And what will end up happening when you're writing a paper, especially those of you that end up sticking with APA, you may have to go back to your reference guides for kind of unique cases or quick reminders, but the general format will be cemented, and you'll find that writing your papers becomes much easier because you don't have to think about that stuff. You're just kind of going through and making minor edits. And so that's the goal. What I'll end up doing, you have that sheet. Once we have a chance to talk through it, I'll put up the answer so you'll have a chance to check your work. It's not graded, so even, I hate saying this because it kind of opens the door, but even if you don't do it, I won't know. If you don't do it diligently, I won't know. So there's no point in that. There's no point in looking at the answer sheet when it's up and filling it in because I'm not checking it. However, having the opportunity to practice means that when it comes to your turn-in papers, so the EP mostly, I have very little sympathy for mistakes in style, if that makes sense. Okay, that's enough pontificating. I think, well, that's the whole day. This whole day is me going on an illness-rattled tirade, and you guys get to listen. Next thing. Oh, yes, yeah.
Just to clarify, the EP, is that like our separate project?
Yeah.
Project?
Right. So this is the individual project. And even though we're starting it now, I don't want anyone to become worried about it. Because we've had our meeting with Lisa now. Even though that was specific to our group assignment, hopefully you found it informative or at least know who now Lisa Brunick is and you feel comfortable going to her. But we'll work through the first worksheet, which is just getting a broad topic and figuring out what you're interested in. And then later, some weeks later, we do our second worksheet. And this is all before implementation, the idea being that we can kind of ease into it. Because what I don't want to have happen is I don't want to get towards the end part of the semester and you have all these things due, because what ends up happening is that every professor on campus assigns big projects that are all due at the exact same time, and there's usually an exam or two sprinkled in there for good measure. And so I want to spread this stuff out enough that it's not overwhelming to you. So, with that being said, you're welcome to, if you wanted to look at your worksheet now, it's up. It's all that one packet, but if you look at the worksheet one part, you're welcome to start tooling through your textbook and figuring out what it is you're interested in doing. You're more than welcome. The only purpose for the in-class work days is because I think it would be wrong of me to not give some class time to let you work on that sort of thing, right? To require it, all outside of class. And so, there we are. Any other questions? No? I was going to say something. Oh, anybody loan me a blank sheet of paper? One person. Okay. If you'll number it 1 and then add your name and pass it around. Because I'm ill, I'm trying not to touch anything that you're going to touch. And so this will be our attendance sheet for the day. We just got the first round of who's here and who's not. Questions, admin, so thank you for filling those out. Okay. So we're going to start, again, we're going to start with what we didn't cover the first day, and it's going to be basic stuff. If you think that we're kind of flying past some of the information, it's because we're going to cover all of it in depth. It's just to get us all on the same sheet of music, as it were, right? So we all know what it is that we're talking about. And so the notes from the first sheet start with talking about what a developmentalist is, right? And so we said that that an individual that studies the lifespan from conception through death would broadly be called a developmentalist. And so developmentalists study how both biological and environmental factors affect behavior, and more than that, more than just behavior. Certainly, behavior in this sense is used to indicate kind of the broad spectrum of what it is to be human, right? And what you'll find is that even though this is true of people who study the lifespan, you're all, I'm sure, familiar with the subdivisions within academia. And so you'll often find people who could be broadly categorized as developmentalists in their tiny kind of clique communities, right? And so depending on what level of analysis they're studying at, whether they're studying at the genetic level or at the behavioral level, depending on what age range they're studying, depending on what species they're studying. And we'll see all of that. So you'll notice that, and I'll write this on the board for you, that you have a blank there and it's asking for the domains. And the thing is, with the exception of the faith domain, these will all be familiar because they're the same ones that we have for our research report assignment, right? So the domains for lifespan, when developmentalists are thinking about what they want to study, are cognitive, physical, personality, and social, right? Not necessarily in that order. So chalk. I was very excited about the colored chalk, but it's not very well. I'm going to steal some of my kids' sidewalk chalk. Maybe that'll be better. So, let's see if we can fix this up. Personalities. Okay. So, these are interconnected. And that's why we talk about, to make it easier to pick a topic when you're going through your report, you look at the domains. Now, for the ages that we're about to talk about, we'll go through each of these domains. What you will see, though, not just in your textbook, but in other books, you'll see social and personality often presented within the same chapter or together. And there's a good reason for that. Especially as we get into early childhood through adolescence, when we're really developing our non-genetic personality, we're also learning how to socialize. We're spending a lot of time with peers, and so it makes sense that those will fit in together. So, lifespan, so from conception till death, is much too broad a topic for any one researcher to tackle. And it should be noted, even though this course is offered through Psych, lifespan is a interdisciplinary subject. And so if you wonder when we're going through the textbook why we're not staying simply with psychological whatever, that's the reason why. And basically what will happen is someone who's interested in some aspect of the lifespan will pick a domain and then they'll pick an age range. But it's vital to realize that the age ranges are not real. It's a social construction, right? And this should make sense as we go through it. So, social construction, as you can see in your notes, is something that's simply agreed upon by society, right? So a good example is we say in our society, when you turn 18, you're an adult. Right? The votes to do all these things. But it's also true in other societies that happens when you're 13. There's nothing in nature that says that 18 equals adult, right? Nothing at all. And so that's what we mean by these age ranges. So what we have to do is, as scientists or as practitioners or as consumers of research, we have to come together and say, okay, how are we going to define this? That way, when I'm talking to you and you're talking to me, we are not talking past each other. It all makes sense, right? And that's where we start with these age ranges. So your first age range is the prenatal period, and that goes from conception until birth. Your second one is infancy and toddlerhood. And you can imagine it starts at birth and that continues until age 3. And then your preschool period generally goes from 3 to 6. Now, one thing that you'll notice is that in many other texts that deal with, especially that age range, the preschool period, you'll hear it described as early childhood. Both are equally okay. These, however, are from our textbook, so that helps us, right? So we have preschool. We have middle childhood. This is from 6 to 12. And you'll notice something before we get further. You're going to notice there's more overlap here than what you would expect if you're doing ranges, right? And that's because those boundaries are really fuzzy. So if you've ever been around children that are right in, say, this infancy and toddlerhood, but getting ready to go in the preschool period, so right around age 3, many places will base it upon their milestones, whether they move into that preschool class, or are they potty trained, can they hop with both feet, can they do all these things. And so for some children that happens right at 3, for some other children it happens at 3.5. Those are all within what is normative, right? And of course, we have adolescents. And this is from 12 to 20. Now, caveat for adolescents is that we've already said this is all a social construction. And you'll notice in your notes that we talk about emerging adulthood as well, because after this we have early adulthood. And then we have a gentleman that comes along, Arnett, that says, well, maybe we should use this emerging adulthood to cover from late teens through mid-20s. And in my research, I use terms called subadult and prime adult. And sometimes I'll use adolescence to go from around 10 to 11 to 25. Now, of course, these are meant to be precise terms, so they're not used in the same way that you might call your little sister or tell your little sister that she's being adolescent. Right? There's no value to it. But we have to define why we're saying that this is the age range. So when you see emerging adulthood, often it's a recognition that Western societal convention is such that you start getting more responsibilities in late teens, but you're not completely independent until a little bit later on, right? So you may still find yourself back at your parents' house or whatever it might be. When I divide up my groups, it's based on neuronal maturation. So how much gray matter has pruned away, and this is starting to happen in adolescence, and how much white matter has accumulated, and that's how I decide. So you've, I'm sure, seen that the prefrontal cortex doesn't fully mature until around 25. Myelination goes until the early 30s, we think now. And so that's how I make that division. The important thing is it's operationalized. It's clearly defined. So anybody reading my stuff can look at it, and there's no doubt exactly who I'm talking about. The other thing is the environment can come in and change these a little bit. And we'll see that when we get into history graded and individual graded environmental factors. But depending on what the environmental impacts are, you may meet milestones earlier or later. If we're dealing with brain development, the reason why I include no females in my studies is because female brains mature too quickly. So if I'm trying to study some defect in this area, if I include women in my study, well, their brains are already matured past that point, and so now my data's all muddy. But it's the exact reason that, especially for the guys, that around that age, one guy looks to another guy and says, hey, watch this. And then goes off and does something ridiculous, like bungee jumping off a roof with the tip of a pine tree or something. Because that good idea and it's triggering a reward center isn't being sufficiently suppressed. Now, all of the ladies that are standing at the base of the house going, that's really dumb, well, that's because they're able to suppress that. This is broadly speaking, of course. I'm sure there are plenty of young women out there that will happily jump off that roof too. So we have young adulthood. Then, of course, you can imagine that we go to middle adulthood, and then we end with late adulthood. So this is and then let's see, middle, everybody see this? Middle adulthood is from 40 to 65. And you can imagine late is from 65 until death. And I think that's it for... Any questions about any of this? Now, when you're doing your projects, your group or your individual projects, of course, you need to focus somewhere in here. If you're looking to your book for inspiration, that makes sense, right? There's you don't want to pick an intervention to improve some cognitive ability that only really makes sense for a toddler, right? So it will be no surprise to me that you come and say, I did this intervention so that I would have theory of mind and know that if someone moves someone else's doll, that person returns, they will not know that the doll is moved. It's assumed, right? No? You're especially you're like, no. No? I guess also. Okay. So, it's okay, guys. I know it's the end of the day, and here I am, sniffling and sweaty and trying to make it through what we have to do, and you're bearing the brunt of that. So, switching over a little bit, we talk about some of the linkages. The main point to talking about linkages is that what I suspect you're going to notice as we go through the semester, and especially those of you who have taken other psych courses. For those of you on the biochem side, this probably won't be new information. Those of you to the, I guess we would say to the other side of psych, maybe it will. But when I talk about evolution being, by natural selection, being the linkage between the two, hopefully when we look at the theory and all the different theories, we're going to talk about which one's right and why, and we'll end up coming up with an answer, well, depends, and none of them all the time, right? And so what we're going to try and do is bridge that gap and show how all these things are related, which I think is really important before we get into conception, right? It's important to understand something about genes and amino acids and proteins before we start talking about these things, right? And understanding why these genes are expressed the way they are. And so we'll get to all of that. So we're going to switch a little bit and talk about influences on human development. Now, again, we don't have to worry too much because I'm going to provide all of these, but you'll notice that the very first thing is we talk about cohort effects. Now, it should be no surprise that there are words that are used in the scientific community that are used very differently, kind of in everyday life. One of those is cohort. So when you talk about a cohort scientifically, you're talking about a group of people that were all born at the same time, right? And so, if you're doing a cohort study, let's say you're doing an observational study or a good example, and I don't know if you would have seen this or not, but there's a documentary series called 7 Up. Have any of you been forced to watch that? No? Oh, that sucks. So, yeah, I think it's on Netflix, maybe some of it. Basically, it's a British documentary that started way back when, I think maybe in the 60s or 70s or something. And they follow this group of kids who were all the same age. And so they start by interviewing them, talking about their hopes and dreams and all this other stuff. And every 7 years, they follow up with them. And so you get to watch them develop, and it's fairly neat. But they would be considered a cohort. I see now that example is lost. I'll have to find a new one. What would a new one be? Is there anything relevant to you guys that would fit that bill? No? Okay. Fair enough. Okay, so cohort effects are examples of history-graded influences. And so what you can imagine is an event that happens very specifically or pointedly within a historical context. And you'll notice on the sheet I talk about hunger winter. It's because I wrote these notes up quite a while ago. And now we have a much better example. So you can probably think of maybe some historical event that's going to affect a bunch of different young cohorts, either in utero or in early life, right? It's happened just now. Anybody? Swirls in this direction. Texas.
Hurricane Harvey.
There you go, hurricane. So you can imagine that all the children that are either in utero during this event, and so Why would that be important if they're in utero? We certainly would expect some increase in stress hormones, so increased cortisol levels over some extended period of time. But also, there may be some problems. Not everyone's stocked up on food, all these things. So you may have some other effects, whether it's through some mini famine event or mini starvation event, or ill effects of water. So if sewage is backing up into the streets, all these. And of course, I'm just making these examples up, but you can see that this would all have an impact on this group of people. Now, a similar group of children in South Dakota may be emotionally affected to some degree, but they won't have those effects. They didn't go through the event, right? The hunger winter example, which, because we have a hurricane and it's happening right now, or it happened right now, it's easier to keep in mind. But that example was basically something somewhat similar. It was a siege during World War II. And basically what happened was there was a famine event, and it was found that it affected the children that were either had just been born or were in utero at the time throughout their lives, and affected their children as well through epigenetic influence. Does everybody understand epigenetics? No, that's a stupid question. There are stupid questions, that was one of them. Has everyone heard of epigenetics? Okay. Anyone not? Okay, so easy stuff, because here's the thing, you will hear it thrown around a lot, epigenetics, without any real attempt at explaining what it is. But basically, this is the most simplistic explanation that I've ever heard, and I like to use it. When you're not all of your genes are expressed. So when you think about expressed, the analogy is getting turned on, right? So a gene gets turned on, and we have 20 possible amino acids and turns into proteins and so on and so forth. Now, what you can think of when you think of epigenetics is that you have a gene that's switched off, so it's not expressing, it's not really doing anything, right? And imagine that as a teacup with the tea bag in it, but nothing else, just kind of sitting there. The hot water is the environmental stimulus, the trigger event. So the hot water causes the tea bag to steep, right? But if you just set a teacup on your counter and put the tea bag in there, in the absence of moisture, it could sit there forever and nothing would happen, right? It would just kind of sit there. And that's the same thing. So you could have a gene that you have, that you've coded for, but isn't expressing, right? But then you have this event, whatever the event is, and that causes the expression of a gene, which then can be transmitted to offspring and on down forward. Now, we'll get more in depth with it, but if you think about that, if every time you hear epigenetics, you think of tea bag in the teacup and hot water being that environmental influence, you'll be okay. It'll start to make a little bit of sense, right? And I think that certainly good to know more, but I think that if you came away with that being your understanding of epigenetics, I think you'd be okay as well, if that's not the area that you intend to go into. Okay, so we talked about cohort effects. Now, the next bullet point is for age-graded influences. So that's one of the blanks. You'll see number of examples just below that. But the heading should be age-graded influences. And those are things that happen no matter who you are or where you live, right? Hurricanes, no hurricanes, you born in the U.S., born in Norway, born in wherever, no matter what happens, somewhere between 10 and 13, you're going to hit puberty, regardless, right? Your brain is going to mature in an inverted U, no matter who you are or where you are. If you are a member of this species, it's going to happen. And if you're a mammal, it's going to happen. And it's going to happen in a similar fashion. So it doesn't matter, right? And so those are events that we can talk about across our species, and they're important, right? Because if we're talking about pubertal maturation, we ought to be able to say something about humans in general and not just humans in South Dakota. That make sense? And so they may not seem important just listing them off, but certainly when we start going through the age ranges, we'll see things that are important simply because everyone in our species experiences them to some degree or another. Certainly, there are gender-based exceptions, right? Some of us are more likely to get prostate cancer than others. We don't, incidentally, there's no Susan B. Komen for the cure of prostate cancer, right? There's no,
That's funny.
very upsetting to me. Okay. So, and we're going to go through all of these. And when we talk about menopause, not only are we going to talk about female menopause, but there is some evidence that there is an age-graded change in males that can be, it's similar, so it's called male menopause often because menopause was already taken. So that's what you do, right? And so we'll be able to, this is fairly new data. We'll be able to talk a little bit about that with these age-graded changes as you get older. The last one, non-normative life events. So the example there is a bit grotesque, but I think it illustrates the point, right? So a non-normative life event I put down, that would be as if you went to the state fair and your entire family died on the teacup ride. Now, aside from any questions you might have about how the adults got on the teacup ride or any of those other things, imagine that's not very common. And so we will see those on occasion where we have these big effects for events that wouldn't affect a cohort and certainly aren't age graded. They're these non-normative events. And often they're translated into case studies. Which, actually, before we get into that, so for key issues, in that survey, the pre-course survey, there were a lot of questions about nature versus nurture. We're going to spend a lot of time on that. So I don't want to seem flippant about my answer now. But the answer is this, and I promise I will convince you of this before you leave. The answer is not today, but in December. The answer is both. If you encounter someone who says it's nature or it's nurture, then they're either ignorant of the science or they're leading you on. It's only really the two answers. Because it is both. We know, especially through epigenetics, we know a lot about environmental influences on not only gene expression but behavior. And certainly it would be silly to say that we're not affected by our biological past. So that could be everything from taking distal things and explaining proximal adaptive traits based on these distal events. So when we talk about distal events, we're not talking about 10 years ago. We're talking on the order of 50,000 years ago kind of thing. And we'll get to all of that. Same thing for most of these dichotomies are false. So continuous change versus discontinuous change. I'll make this make sense by giving you one stupid example. You would, I suspect, agree with me, that nobody goes to sleep an adolescent and wakes up the next morning an adult, regardless of the construct, right? It just doesn't happen. And so what we see is this gradual, continual change. However, we know there is some discontinuous change, right? Growth spurts. It's an easy one. Someone goes through a growth spurt. They're not growth spurting the whole time, or else you would have Andre the Giant. And there are pathologies that are not named for him, but that can explain that. And so you do see this case of both are true. You do have gradual change, and the gradual change when you look at cognition, so a gradual increase in cognition, and then we see a gradual decrease starting in the late 30s in cognitive ability going until death. We're going to talk about all of that stuff, but it's also true that we have these punctuated events where we get better at some things. Okay. And the same is true for critical versus sensitive periods. We're going to talk, when we get into the prenatal period, mostly in the prenatal period, we're going to talk a lot about terogens and these toxic things that can affect fetal development. And I think that we'll also talk about organ function. And so the reason why those two things are important, which I guess I should explain now, is typically, not always, because certainly there could be some genetic issue. But typically, if there's some malfunction of organ development or organ function, there's a teratogen that's influencing that, whatever it is. So whether it's alcohol or cigarettes, whatever it might be. And we'll go through all of those. But it is true that there are periods where it's absolutely critical. So you'll see early experiments in vision used cats. Cats have phenomenal eyesight. So they used cats, and one of the things that was done was that cats' eyes were covered, like one eye versus the other, and in some experiments, kitten was either placed into a chamber that had only vertical lines or only horizontal lines. The object was to see if that affected their vision later in the life course, and certainly it did. So, and we'll get further into this. I don't want to go too deep today. But neurons don't fire to a whole picture. You have individual neurons firing to different shapes and lines, right? So different orientations. And so cats that were only exposed to the vertical lines didn't fire at all when they saw horizontal line. And cats that had an eye covered or had their eyes covered during the critical period never developed sight. And we'll even see that with human children. Human children that are not exposed to language in any way. So they're not exposed to verbal language, or they're not exposed to sign language, if that happens to be their case, will, after a period of time, lose the ability for language. And so we could call those critical periods, right? But there are also sensitive periods where you're more susceptible, but if there's a deficit, we can recover it, and we'll talk about those. So, for our remaining time, which isn't all that much, we'll go through the basics. So, the basics of research methods. You've either taken research methods or you're going to have to. This isn't a research methods course, so we just want some base level understanding. The first thing we have to do is have some sense of scientific process and the scientific method. And so, if I can find this. You guys, everybody here knows who Richard Feynman is, right? Or was. He's very distinctly dead. Richard Feynman was a physicist and more importantly, a really good communicator of science. And that's a key difference, because I suspect all of you have experienced this, that you go to some family event and crazy Uncle Bob is there, and crazy Uncle Bob says, "Tell me what you're doing in school." And even though it makes perfect sense to you, you can't explain it in a way that makes sense to crazy Uncle Bob. It's not because Uncle Bob is crazy, it's that there is a difference. I actually do have a crazy Uncle Bob, and I love him. Now. The fact that you guys are snickering is rude, and I don't appreciate it because Uncle Bob is awesome. But the point is, there is a gulf between understanding scientific concepts and being able to explain them. And that's why we hold up good explainers of science. That's why people like Neil deGrasse Tyson are so popular, right? Or Carl Sagan. And you can go on down the list. But Feynman was a great science communicator, and this is basically what he said. So he was giving a lecture in 1964 on this topic, and you can watch it on YouTube, actually. It's good if you don't learn black and white. And basically what he said, and I've used this ever since, well, not since 64, I'm not that old, but said basically what you do is you start with a guess. And you compute the consequences. And then you observe, you compare with nature, so you observe nature. Now, this is going to seem crude, right? But it is important, and we do start with a guess. It's an educated guess, but often it's a guess. And when I say educated guess, it's as you go through your academic career, you're accumulating knowledge, right? And so often the guess takes this form, depending on what discipline you're in. Given what we know, we, the scientific community at large, given what we know, this thing should be true, whatever this thing is. And so you compute the consequences. That's the probability, the likelihood of it being true versus not being true. So if you think about something like UFOs abducting people, what's the probability of that being true versus not being true, right? You can do that on your own. But this is where we're dealing with hypotheses, right? So we make a prediction, we compute the probability, and then we compare it with nature. Now that can be naturalistic observations, so ethnographies, things like that. More likely experiments. Experiment is the gold standard. Now, this process is not, it's not an individual process. Nobody gives a crap what I think about anything. And that's the way it ought to be. So, when I do this, then it's on the next lab to say, okay, well, let's replicate that. Let's try and replicate that. If my results, when I compare it with nature, I do the experiment, if my guess is wrong, then the guess is wrong, period. It's done. Doesn't matter how nice it sounds, how warm and fuzzy it makes me, it's wrong. Now, if all of this agrees, and then the next lab that replicates, and they don't come up with that conclusion, and then the one following that doesn't, then we need to get rid of the theory, whatever this first guess was. And that has to be that way. And most people are more than happy to do that. Because science doesn't have authorities, so nobody is immune to having their theories tested, right? And because science doesn't have the authorities, one, we want to advance. We know that we're trying to get to some approximation of truth. And so we want that advance to happen. So if someone overturns your theory, then good on them. And certainly that's how you make your name, right? You prove someone else, you disprove someone else's theory. And so here we have our basic setup. Now, of course, the scientific method, as you know, is couched in theories and hypotheses. And I suspect you all know that theory is used very differently in this context than it's used everywhere else, right? So you're talking with friends, you might have a theory about something, and that is to say that you have an idea about something or how something works. That's certainly not how we use theory in science. Theory of relativity is not someone's best guess at the bending of space and time, right? And so it's used a little differently. And one of the ways you can tell, there are a lot of nuances, one of the ways you can tell is that a scientific theory will have testable and falsifiable hypotheses. And so when you're dealing with pseudoscience, what you'll find is that there's no real way to test the theory. They're usually vague enough that no matter how the chips fall, that person can claim victory. So, within the scientific context, I'm going to make definite predictions. I'm going to say, based upon what I know, based upon my theory, whatever it is, I think that X should be true. So I'm going to have what's assumed. It's usually not written out. My null hypothesis, there is no difference. And then I'm going to have my predictions. So I think that exercising 3 times a week will improve cognition as measured on such and such test, right? And I'm going to say an improvement is this margin, right? So I can falsify that. If it doesn't happen, it doesn't happen. And I need to either modify or throw out my theory. Maybe I need to modify it a little bit, or maybe I need to modify my testing. But we can know something about that. If you drive down Minnesota Avenue and you stop by the Sioux Falls Psychic office and you say, well, how does this work? Or you go to an acupuncture clinic and you say, really, chi? What is that? How can you measure it? You will never get a definite answer. You'll never get anything that is testable, ever. Now, there is evidence that you can stick needles in people and there's some therapeutic effect, but it has nothing to do with any specific place at all, right? Because this thing doesn't exist. You see. Okay. Does that make sense? A little soapboxy, but there you go. Okay. Of course, science doesn't prove things. It's not our goal. You could not come up with enough tests to prove that, I don't know, gnomes lived in your yard. You can say that based upon the evidence that I've collected, it's very unlikely. Probability is very low, and it's low enough that it would be absurd to say that there are gnomes, or there might be gnomes. But you certainly, you couldn't prove that. And that's okay. That's not a problem. And as young scientists, you have to get comfortable with that. You have to be very comfortable with saying, either we don't know yet, jury's still out, or we don't know, or yes, we can't prove it, because often you'll have people come up to you, maybe you've gone to a high school reunion already, and someone's come up to you and they say, prove it. And you say, well, proof is either a philosophical or a mathematical term, and it's talking about a logical progression of an argument. It has nothing to do with how it's used kind of in the lay public. But also, we don't do that. We can disprove definite hypotheses, right? Or we can test hypotheses, can break down a theory. So we can do that. We can say, I don't want evidence for X, Y, or Z. We don't prove it. GIGO, which is the next line down, it simply means garbage in, garbage out. I suspect, so you'll hear this throughout science departments that you're in. You'll hear someone say GIGO. And it's basically the more professional way to say probably what your grandparents said, which is something to the effect of, don't half-ass it. If it's a job worth doing, it's a job worth doing right, or some such thing, right? And so we want to be very skeptical in general, but we want to be especially skeptical if we encounter vagueness in someone's theory. Now, the reason why I'm spending any amount of time on this is because soon enough you're going to strike out on your individual project, and you're going to have to come up with some intervention. And you're going to have to, even though you're going to lit, you're going to have some tools to be able to weed out the BS from the legitimate research, right? Because maybe in your idea formation, you're not going straight to the lit. Maybe you're looking around. And so I think between the baloney detection kit and what you generally know about good science, you should be safeguarded in that matter. So for critical and clear thinking, the key then is to be skeptical but not cynical. There's nothing wrong. You shouldn't believe anything I say just because I've said it, right? Everything is open to question. Now, it's true that we can't know everything, so we have to rely on authority to some degree, but that doesn't mean it's not open to question. So if someone tells you that they just gave away their cow for magic beans, you should have some questions. You shouldn't let them slide with that. Well, okay, well, what do you mean by magic? Where did you get them? What do they do? How do they do it, right? All of those things. Before we go, I'm going to quickly get into the categories of lifespan research. And I'm gonna say this. So your textbook divides up correlational from experimental research. We talk about correlational research in the context of naturalistic observation, case studies, surveys, things like that, right? So naturalistic observations, typically those are ethnographies. So this is something that came over from anthropology, kind of out there observing people doing their thing, but in most cases not intervening in the situation. Case studies are typically in the medical sense. Often they're very rare cases. So those non-normative life events, family dying in the teacup ride, you're not going to find a representative sample of teacup family deaths. So You might do a case study, or the population is extremely vulnerable. Now, before you head out, one thing I want to make clear, because I don't think it was clear in your textbook, is that just after correlational research, the textbook talks about EEG and fMRI and CAT scans, right? And it may not make sense immediately. The reason why I think it's where it is because those are correlational methods used experimentally. So let me explain that quickly. And I happen to do EEGs and fMRIs, so yippee for everyone. When we do an fMRI, what we're measuring is blood flow or blood oxygenation, right? So you're looking at the transfer from oxyheme to deoxyheme. You're not measuring neurotransmitter release, right? You're not measuring neuronal firing. What you're looking at is a correlation. So our assumption is the more that brain cells fire, the more food they need, more energy they need, and so the more blood that's going to go to that area, right? So that's a basic assumption. And so when we see that, that's the activity we're seeing, but it's a correlation. It's not the actual activity. EEG is a little different, but EEG, the action potential from a pyramidal neuron, from a postsynaptic potential on a pyramidal neuron, is very small. And you have to pick it up through skull and everything like that, right? And so what you're measuring is this summed potential, so all of the hundreds of thousands of neurons firing in response to that event. And you're making some assumptions. That's why that's correlational, but they are used experimentally. You will not find a correlational study using EEG or fMRI. They're all experimental. Okay, so we're done. Last couple things, remember, room 001 on Friday. You're welcome to bring your computers. You don't have to. I'm going to post the notes. That way everybody has the same thing for all three sections. And if for some reason you need to talk to me, please send me an email. I'm not trying to blow people off, but I am going to go directly home and try and get better. But anything before I leave?